First episode of the energy unit. In the last unit, which was cells, we did an overview of the organelles in a eukaryotic cell. And then we looked at the cell membrane in more detail, seeing how it regulates what materials can enter and exit the cell. Our current unit will be focusing on the mitochondria, which is how the cell utilizes energy through cellular respiration, and the chloroplast, which is how a plant cell creates energy through photosynthesis. But before we jump into these processes, you need to learn a little bit about free energy in general and how it works in an organism's metabolism. So in this episode, we'll look at the two laws of thermodynamics, what free energy is and how we can use it to describe reactions, and finally an overview of ATP, which is what the cell uses for energy. So let's get started. Hopefully from the last unit, you understand that the cell is very dynamic, with components moving here, there, and everywhere. In order to do everything the cell needs to do, there are thousands of chemical reactions happening in the cell. And the sum of all of these reactions is referred to as the organism's metabolism. All of the chemical reactions taking place in the cell can be organized into a series of biochemical or metabolic pathways. Here's an example of a metabolic pathway. We have a multi-step pathway with a starting molecule and a product. In this particular example, there are 3 reactions being catalyzed by 3 different enzymes. So the product of one reaction becomes the reactant for the next. One of the objectives of this video and the section in the book is to be able to use the correct terminology to describe a reaction. This brings us to our first 2 vocabulary words. If you're breaking down a complex molecule into simpler, smaller molecules, like polymers to monomers, this is described as a catabolic reaction. These pathways will release energy as it breaks down a molecule. In this unit, we'll be studying the pathways of cellular respiration, which is the major catabolic pathway of cells, where glucose is broken down and the energy stored in glucose is converted to ATP, which is the type of energy the cell can use for work. The opposite reaction is where you would build a complex molecule from the smaller, simpler ones, such as monomers to polymers. This type of reaction is described as anabolic, or sometimes referred to as a biosynthetic pathway. These pathways will require energy to make bigger molecules, such as putting together amino acids to make proteins. This unit also looks at the major anabolic pathway of photosynthesis, where we take an input of energy from the sun and along with some water and carbon dioxide, build glucose and other organic compounds to store that energy. We use catabolic and anabolic reactions together to drive the overall pathways of life. Energy given off from the catabolic reactions is used to power the anabolic reactions. So let's quickly review energy before we move on to other ways we can describe reactions. If you remember from your summer assignment, energy is the ability to do work or the capacity to cause change. Energy can exist in various forms, like the ones listed on this slide here, which are the forms of energy that we deal with in biology. And it's important to know that energy can be converted from one form to another. You can pause the video if you want to copy down the terms and definitions, but I'm going to move on. Which brings us to the laws of energy transformation. Talking about how energy can be converted from one form to another is called thermodynamics. You can learn more about thermodynamics in a chemistry class, but I'm just going to discuss what you need to understand for the purpose of biology. The most important thing is that organisms are open systems, so we can transfer energy with our surroundings. There are 4 laws of thermodynamics. However, we're only going to deal with 2 of them. The first law of thermodynamics is pretty easy to understand, and you've probably heard it before. Energy cannot be created or destroyed, but it can be converted into different forms. This is also referred to as the principle of conservation of energy. So looking at this bear, which is the example they give you in your book, as the bear is eating the fish, the chemical energy from the fish is being transferred to the bear. Once upon a time, you learned this in an ecology unit with food webs and trophic levels and which way the energy goes when one organism eats another organism. We're not making energy or getting rid of energy, we're just moving it from one form to another. Which brings us to the second law of thermodynamics. Energy cannot be changed from one form to another without a loss of usable energy. As we transfer energy from one form to another, some of this energy is given off as heat. We haven't destroyed it, It's just part of the environment now. Remember, we can exchange energy with our surroundings because we are open systems. So every time we transfer energy, and some of that energy goes to our surroundings, we are increasing the entropy or the disorder of the universe. So as far as our bear example is concerned, the last time we saw the bear, he or she was eating a fish, and the chemical energy from the fish was being transferred to the bear. Now the bear is transferring that chemical energy into kinetic energy as it's running through the water. As the process of cellular respiration is happening, energy is being given off as heat, while the products of cellular respiration, water and carbon dioxide, are also being exchanged with its surroundings. So as the organism is becoming more ordered, this is increasing the disorder in the surroundings. I know this can be a difficult concept to understand, so let's use a teenager's bedroom to try and illustrate the point. On the left side, we have a neat organized room that has low entropy. However, the room is also seen as less stable because over time the room will have the tendency to become messy. The messy room is disorganized, high entropy, but more stable because without working on it, the room will tend to stay that way. Another term that can be used to describe reactions is spontaneous. If something doesn't need any energy input, then it's said to be a spontaneous reaction, like a neat room going to a messy room. However, if something requires energy, like the work you have to put into making your messy room neat and organized again, then it's said to be non-spontaneous. If you're still confused about the whole entropy concept, don't lose sleep over it. Just understand that as cells organize themselves, the lost energy is given off as heat. So this brings us to the next concept in the chapter. When we're looking at all these reactions that are happening inside our cells, we can categorize them into which don't need energy versus which ones need energy. Another way to say that is which reactions are spontaneous and which are not. The way scientists can do that is to measure the change in free energy, or delta G. The triangle symbol is the Greek letter delta. We don't call it triangle G. And the G stands for Josiah Gibbs, who's the person who developed this concept. Delta also refers to as change, which is why delta G is called the change in free energy. Now, up until this year, this formula was on the formula sheet, which meant that College Board could ask you to calculate Gibbs free energy. Not that these problems were difficult, but this year, the formula is not on the formula sheet, and from what I understand, you probably won't ask to calculate this anymore, so I'm dropping this from my curriculum. However, you do need to know what the number stands for. To summarize, if delta G is negative, then the reaction is releasing energy and is said to be spontaneous. The energy given off can be used or converted to do some other type of work in the cell. If delta G is positive, then that reaction requires energy for it to go to completion. Two more ways to describe a metabolic reaction would be whether the reaction is exergonic or endergonic. An exergonic reaction is one that releases stored potential energy, has a negative delta G, and would be considered spontaneous. Looking at the model, exergonic reactions are where the reactants have more free energy than the products, so energy is given off or released. The reduction in delta G makes it negative. An endergonic reaction would be the opposite. It absorbs energy and stores it, has a positive delta G, and is non-spontaneous. The model shows that endergonic reactions need energy, so the reactants have less energy than the products. The increase in delta G makes it positive. So what does this mean for cells? In a closed system like this one, reactions will eventually reach equilibrium and stop working. But we've already established that organisms cannot reach equilibrium, because if so, it'll stop and the organism will die. But cells are open systems, and with all of the materials constantly moving in and out of the cell, they will never reach equilibrium. That's one of the defining features of life. Metabolism is never at equilibrium. The product of one reaction is not just accumulating, it's being used as the reactant in the next step of the biochemical pathway. This keeps us from ever reaching equilibrium. The last thing we need to go over is how a cell actually does the work it needs to do. We can basically break down cellular work into being one of three types: chemical, such as building molecules; transport, which we saw examples of in the last unit, and mechanical, actually moving parts around the cell. The cell is very efficient at managing energy by coupling or pairing together an exergonic reaction, which releases energy, with an endergonic reaction, which needs energy. And the primary molecule that's involved in energy coupling is adenosine triphosphate, otherwise known as ATP. To remind ourselves of the structure of ATP, we have adenine, which is a nitrogenous base, joined to a ribose molecule, which is a 5-carbon sugar. Together, these two make up the adenosine part of the molecule. Joined to the sugar are 3 phosphate groups, which gives it the triphosphate part of the name. Remember, phosphate groups are functional groups that are used to store and transfer energy. So when the 3rd phosphate group is hydrolyzed, some of the stored potential energy from the molecule is released. The molecule that remains is ADP, or adenosine diphosphate. And this reaction will partner with an endergonic reaction and provide the energy for the endergonic reaction to go. Let's look at an example of chemical work. Here we have an anabolic reaction where we're converting one amino acid, glutamic acid, to another amino acid, glutamine. Overall, this reaction is endergonic with a delta G of positive 3.4 kilocalories per mole. So normally this reaction is not spontaneous and needs energy to go. So this is what happens when we couple this reaction with the hydrolysis of ATP. The phosphate group that's broken off of ATP is transferred to the glutamic acid. This is called phosphorylation, and the molecule is referred to as the phosphorylated intermediate. This phosphorylated intermediate is more reactive now. I usually think of it as being primed and ready to do work. At this point, the ammonia molecule can come in, replace the phosphate group, and covalently bond to glutamic acid, now creating glutamine. And we're left with ADP in the inorganic phosphate group. We've already seen that the delta G for the conversion is positive 3.4 kilocalories per mole, and the delta G for the hydrolysis of ATP is negative 7.3 kilocalories per mole. So by coupling these two reactions together, the difference, or the net delta G, between the two reactions is negative 3.9 kilocalories per mole. And because this overall free energy change is negative, it will happen spontaneously. The same process occurs for the other 2 types of cellular work. For active transport, the phosphate group attaches to the carrier protein, making it the phosphorylated intermediate and giving it the energy it needs to change shape. For the motor protein, the process is a little different. While we don't phosphorylate the motor protein, the ATP will still hydrolyze and cause the motor protein to change shape and move down the microtubule track. The last thing to mention about ATP is that it's a renewable energy source. We've been talking about how ATP can undergo hydrolysis to make ADP in an inorganic phosphate group, which will release energy for cellular work. But ADP in the phosphate can be the recipient of released energy from another reaction to regenerate it back to ATP again. And this cycle can be repeated over and over to constantly store and release energy in order for the cell to do work. I know this was a long episode, and thanks for hanging in there, but in order to understand the pathways that we're going to see in cellular respiration and photosynthesis, you really do need to understand these concepts. So bring any questions to ask during class, and as always, be a good human.